So this is the job of the B and the T cells. Now, I have taken the liberty to draw for you one B and one T cell. They're just lymphocytes. You may have already seen these in lab. So here's a B lymphocyte, here's a T lymphocyte. I can actually draw lymphocytes, as you might recall. Now, there's no way to tell just by looking at a lymphocyte by its appearance, this roughly spherical cell about the same size as an erythrocyte, mostly nucleus with a little hint, a little touch of clearish looking cytoplasm on one side or the other. There's no way to tell if this is a B cell or a T cell or an NK cell or what sort of lymphocyte it is, unless you were to actually watch it do something, unless you watched it perform its function. What we call the activities that B lymphocytes do, that's called antibody-mediated immunity. It's B cells that have antibodies. Yes, I did over-enunciate the B in that word on purpose. B cells make antibodies. They have antibodies. So anything to do with an antibody references B cells or B lymphocytes. T cells carry out what we call cell-mediated immunity. No antibodies involved for T cells. So they perform cell-mediated immunity. Both of these things, both of these activities are part of specific immunity, be it a T cell or a B cell, a T lymphocyte or a B lymphocyte. Specific immunity, you might remember, is specific, meaning specific to a particular pathogen. Not like non-specific that might work against a number of things. Think of those non-specific or innate defenses that you've been learning about. The presence of skin, the complement proteins, interferon proteins, etc. These things might work against 2 or more or 20 different types of pathogens. Specific immunity is only going to work against a specific pathogen. In fact, I will do you one more than that and say it's specific to an antigen, which we're going to talk about in a moment. Specific immunity is also systemic, which means it happens throughout your entire body. If you join me here at A&P Nerd headquarters, which of course is, where we're going to hang out, you and I, A&P Nerd headquarters, you can see the decor behind me. I have put back up my circulatory system poster and the lymphatic system poster, our two vascular systems, just as a visual representation of this systemic nature of specific immunity. Where all these vessels go, that's where the T and the B cells can go. In fact, don't we know, you and I, that the T and the B cells can actually leave the cardiovascular system to go somewhere else. So anywhere that these T and B lymphocytes can be, that's where we can perform specific immunity. Unlike non-specific, which oftentimes is localized. If I have a wound, say here on the back of my hand, and I could have inflammation right here, but my innate or non-specific immune response is only going on right here where I have the wound. Yes, there are a few more systemic-ish parts of non-specific immunity, but specific immunity is systemic, can happen anywhere in your body. This is the type of immunity we say has a memory. It gets better, faster, stronger with repeated exposure. You've already put that into your notes. So again, this is all about the T's and the B's. The T and the B cells, this is what they do for us. Now we need to describe antigens. What are they? An antigen. Something that has the ability to trigger or start a specific immune response. That's the textbook definition of what an antigen is. An antigen, something that has the ability to trigger or to start a specific immune response. Now you'll look at what I created here, a very high-tech visual aid that I made today while I was sitting in my 224 office hour because nobody showed up to ask me a question. And that gets kind of boring. So I created a little visual aid to help us out for a lecture here. And look at the rather cheesy name tags that I put on my two cells. Hello, my name is self cell. And to the right, hello, my name is pathogen. These antigens are chemicals that are found where? On the cell membrane of a cell, on the surface of a pathogen. Not all pathogens have a cell membrane. So on the surface or the cell membrane of some cell, some pathogen, however you might want to say it, that's where these things live. And what they are is cell surface ID molecules. Cell surface identifiers. They are almost always proteins, big, large, complex proteins. Sometimes they're larger glycoproteins, large carbohydrates, great big polysaccharides, even large lipids in a few cases. But for the most part, we're talking about proteins here. Now, who has antigens on their surface is a good question. The rather snarky answer is yes. Everybody has antigens on their surface. Everybody has some sort of protein, we'll say, on its surface identifying this thing for what it is. Are you one of my cells? Are you another cell? Now, I put the great big, sticker name tag like you're at a conference or something, but in reality, we would be talking about some sort of protein sticking off the surface of this cell. This would be an antigen right here. A specific collection of amino acids in a specific order with a specific three-dimensional shape. The pathogen also has the antigen on its surface that identifies it for what it is. You might recall way back in 189 talking about the function of these cell membrane proteins. Weren't some of them doorways or channels, some of them receptors, some of them embedded enzymes like acetylcholinesterase, if you think all the way back to 223. Some were components of the cytoskeleton. Some were ways a cell is connected to another cell, like gap junctions. But some are cell surface identifiers. These are called antigens. So the antigen is the protein on the surface of your cells, my cells, pathogens that identify them for what they are. Again, in almost every case, proteins. And it's these things, these antigens, that might trigger a specific immune response. Now, if you look at my self-antigen over here, this one, this self-antigen identifies this as a Duane cell, let's say. Do you think I normally mount a specific immune response against my own antigens? No, almost never. I could. That would be an autoimmune disorder, wouldn't it? So specific immunity is specific to an antigen. That's what the T and the B cells actually respond to, is a specific antigen. As an example, I have called up this picture. This is actually an indoor beehive. I imagine a dog or a cat would have endless hours of fascination by one of these things. This is a beehive in plexiglass. Right here. Inside a house, see the little tube that goes out the window so the bees can go in and out. Now, the only reason I bring this up as an example for specific immunity is, of course, I have a little analogy that goes with this. This plexiglass beehive that you see right here is very similar to one a friend of mine used in graduate school. So I want you to imagine the wall in a biology lab, and what she had were two plexiglass beehives like this, hive A, hive B, in her lab, connected to the outside on the third floor of the McInerney Science Building at the University of North Carolina, Charlotte. Now, what she would do, of course, is make observations through the glass, watching the little bees. What are you doing? Each bee had a little dot glued on its thorax with a number like red 5. So she would watch red 5 for half an hour and see what red 5 does. Does red 5 do the waggle dance? Does red 5 do a circular dance? What's red 5 doing? To document the behavior of the bees. So one day, I was picking up this friend of mine. I think we were going to have a study session for exams. So my job was to swing by her lab and get her. So here's Dwayne. I swing by. And she says, oh, I've got 5 minutes left on an observation. Can I go ahead and finish that before we leave? I said, sure. Can I watch the bees? She said, yes. So I stood there and looked through the plexiglass, watching the bees. And my attention was drawn to the door here that led outside, straight out into the air on the third floor. Because as I looked at the entrance to the beehive, I saw great big honeybees, bigger than everybody else. And I asked my friend, what's with these bees standing here at the door? That's what these actually are. And she said, oh, those are the guard bees. I said, what are they guarding? She said, oh, they're guarding the hive to keep out intruders, robber bees, ants that might try to come in, and so on. And then she said, hey, keep watching, because you know what we did, Dwayne? I said, what? She said, we put the two hives too close together. Their doors aren't far enough apart, and sometimes the bees make mistakes. Keep watching. So I said, all right, I'm game. So I keep watching the guard bees. So here comes a little bee, flies into the door like this, and lands, and then she starts walking into the hive. The guard bees run over and start beating on her with their antennas. Whack, whack, whack. Hit her with their antennas. Like that. And then they step back and let her pass. In she goes. Next bee comes up. Same thing. They run over, beat her all over the body with their antennas, let her go. And I keep watching, wondering what's going to happen. Well, sure enough, a bee that was supposed to fly into hive B accidentally went into the wrong door. She went into the door for hive A while I was watching. And certainly the guard bees walk over to her, beat on her with their antennas, and then one of the guard bees took its front legs and just slammed this little worker bee to the floor, all these other bees started coming over and they stung her a bunch of times till she was dead. Then they drug her over to the edge and they threw her off the building from the third floor. Down she went, down to the ground dead. Now, that is a rather harsh punishment for going through the wrong door, certainly. But here's my question for you related to antigens. How is it that these guard bees recognized the bee from the wrong hive? How did they know she wasn't supposed to be in A? She smelled bad, or smelled wrong, smelled different. She did not have the pheromone or chemical markers that identified her as a bee from our hive. She's an intruder, maybe going to kill the queen. We've got to stop her. This is the job of the guard bees. They've only got one job, and man, do they do it. This relates to the nature of antigens. These proteins that are on the surface of your cells, my cells, everybody else's cells, and on the surface of pathogens are just like these name tags, and they identify what we're looking at as one of us or not one of us. That's what they do. And we are specific to the antigen. So specific immunity hinges on the ability to recognize these antigens, these cell surface proteins, as self or non-self, as one of my cells or a cell from somewhere else. That's the job of these antigens. Let me zoom in super close up on the antigen protein on the surface of my fictional pathogen. So it's not the whole antigen that triggers an immune response. Maybe it's only this little section right here, or maybe it's this little section right here. That's called the antigenic determinant. The antigenic determinant, or some people like a one-word name for that. They call it the epitope. This is the very specific section of an antigen protein that triggers the response. So the specific or the small part, sorry about my pen there, small part of the antigen that actually triggers the response, is called the antigenic determinant or epitope, synonyms for one another. So it's not even the entire protein, it's just one specific part of the protein that these T and B cells can respond to. Now remember, I'm going to say this again. Everybody's got antigens. You just don't normally respond to your own. But what if we took this self cell, this Dwayne cell, let's say it was, I don't know, on one of my erythrocytes. Maybe it's on my kidney cells. If I gave you a transfusion of my blood, or I gave you one of my kidneys, are you necessarily going to accept that with no tissue rejection? You might respond to my antigens as if they are pathogenic, right? Don't we sometimes reject a tissue transplant? But if I give myself a tissue transplant, if I squeeze out a pint of blood today and I give it to myself in a couple of weeks, I'm not going to reject that because I'm giving myself self-antigens. So when we do a tissue transplant, we looked for cells that have antigens very similar to mine, as close as we can get without cloning them. Isn't this why we typically look for close relatives to be tissue donors first, for a bone marrow transplant, a kidney transplant, and so on? If I've got an immediate family member, I'm going to test them first to see if they'll work, because maybe their antigens are a little more like mine than yours are like mine. So this is very, very specific what we're talking about here. Specific to a protein of particular size and shape. Let's go past my little beehives here and let me draw for you, again, those two varieties of lymphocyte that we're talking about. The B lymphocyte and the T lymphocyte. B cells, B lymphocytes, are a little bit different than T lymphocytes with regard to what's on their cell membrane. So here's what I'm going to do. On the surface of the B cell, I'm going to draw this whole bunch of Y-shaped proteins. These are antibodies right here. That's what I have on the surface of a B lymphocyte. On the T cell, I always make them sort of curly cues just so they look nothing like the antibodies. On the T lymphocyte, we have what are called antigen receptors. Antigen receptors right here. This B lymphocyte has antibodies. That's what we call these proteins. They're proteins. So are the antigen receptors. For us humans, these are usually always proteins. So this B cell, this individual, let's say Tom. Well, I guess it should be a B. So let's say Barb the B cell, and Tom the T cell. How's that? Every antibody on the surface of Barb the B cell is identical to every other one. And they can only respond to one specific antigen. Or I could say one specific antigenic determinant. The same thing with the antigen receptors on Tom. Only can respond to one specific antigen. Let's put it this way for you, maybe. So think of a little pathogen, a bacterium, a virus, whatever you want to think of, with its own antigens on it, okay? Here's how it works, very cut and dried. See this antibody on the B cell? It either fits with that antigen or it does not. Same thing with this antigen receptor. It either fits with this specific antigen or it does not. That's the kind of specificity that we're talking about. And when you think about these molecules, everyone, don't think of them in two dimensions. The only way that I can draw them here. You need to think of these things three-dimensionally. Everybody talks about proteins fitting together like a lock and a key, don't they? But that's not enough dimensions for me. Oh, sure, it would work like a lock and a key if the key had about, 270 rows of teeth on it. And then the lock had about 270 receptacles for teeth. Because what I'm telling you is this. If you think of this antigen on my red pathogen there, or the antigen receptors on the T lymphocyte, these are three-dimensional proteins. The same thing with antibodies. So these cells have on their surface these proteins. So if you think of an antibody or an antigen receptor, think of it like a trough, like the snowboarders and skateboarders would say, a half pipe. All sorts of three-dimensional shaping in this half pipe because of what? All the amino acids and the way they're bound together. So unless the antigen fits into this trough exactly right, the two don't go together. So the antigen has to fit like a three-dimensional key into a lock, which is the antibody or the antigen receptor, or we don't have a response. What I'm trying to do is really illustrate to you the specificity that we see in this interaction between antigens and antibodies on a B cell or antigen receptors on a T cell. And just for you people that have to take micro as well, a lot of microbiology professors will call these B cell antibodies, the antibodies on their surface, these things right here, they'll call these things antigen receptors also. You can if you want to, but guess what? They're antibodies. We'll see in a little bit. So these things are actually antibodies on the surface here. That's what they are. These are antibodies. And they would all be, whoa, that's a little mess there, isn't it? They would all be these antibodies designed for, programmed for the same exact antigenic shape. Same thing with these antigen receptors on the T lymphocyte. This T lymphocyte can only react to one specific antigen. Has to come across it. So, to not put too fine a point on it, if I think of you, or me for that matter, so here you are, a normal, healthy individual. Look, you're all happy, right? That's you or me. So you're very happy right now. You have in your body right now as an adult, literally millions and millions of different B cells, each one programmed for a different antigen. Millions of different varieties of B cells. And they're either going to run into their antigen at some point throughout your life, or they are not. So these T's and B's are genetically programmed as to what receptor they'll have on their surface. And you, again, literally have millions upon millions of different B varieties and different T varieties. But not to scare you in this discussion of immunity, the number of different T and B's that you have is finite. I'm going to say that again. The number of different T's and B's you have is finite. Thank you, Mom and Dad. So when a pathogen gets into us, into one of us, let's say this pathogen right here, I get it. I either have B and T cells programmed for it, and I'll have a specific immune response, or I do not, and I won't. It's really, really like that. So these T's and B's show high specificity for what they will do and what they can do.